Good afternoon. My name is Zdravko Petanjek. I am professor of anatomy and neuroscience at the School of Medicine, University of Zagreb. I wish to thank to have opportunity to present my results. Uh, my main focus of research is related with development of specific classes of neurons within prefrontal cortex. As you can see on this picture, prefrontal cortex is heavily expanding region during prima development. It's very discrete in other non-primate species and is important to processing some of the most complex cognitive functions. Specific features of the human cortex is that in the layer 2 and 3, which are the source, which are the place of origin for the corticocortical projections, there is increase in proportion of neurons. So what is characteristic of the human cortex is that they have a exponential increase in number of corticocortical projections, which is the prerequisite to allow human to process human cognitive functioning. However, when you have such network expanding in numbers, you have to make also the proper integration. So there might be some population of neurons which have particularly important role to establish proper integration and better processing through this corticocortical network. So when we use some neurofilament antibodies, you can see when comparing the monkey with the human that there is increase in population of corticocortical neurons marked showing heavy reactivity, and not only that they are more densely packed in human, they are also much larger in human. So there is several times larger increase in number of specific population of neurons than is the overall increase in number of corticocortical neurons. So, as I mentioned, our primary goal is to study their development and to connect how this relates with the development of the cognitive functions, how this might be altered in different mental and cognitive diseases. But we are also interested to see how these neurons are reacting during aging. So what is already known from the literature, that this reactivity, so this is a layer 3, this is the layer 5, so layer 5 is projecting to subcortical structure. It contains also neurofilament stained neurons, and you can see that this layer is not so much affected in the Alzheimer's disease like it's the layer 3C, so these neurons which are important for the integration. So we might conclude that if these neurons are, for example, selectively affected, you will have much more consequences, much higher consequences on the cognitive processing than if this at the same way will other population of the neurons will be affected. And also we can look in the opposite way. So if these neurons are sparred and you have much decrease or atrophy in other population of the neurons, that cognitive function might be preserved much more than you might assume when you are looking to the structural changes to the, for example, brain atrophy. There is also these neurons can be marked by acetylcholinesterase, and as they appear with this heavy staining during early childhood, they also already is known in the literature that they might be selectively altered uh, um, during the aging. So what they are doing, so they establish rich intracortical connection, and at the same time they connect several cortical areas. So meaning by this rich intracortical connection, they can control the whole output of the prefrontal cortex and changing the whole functional properties of the prefrontal cortex. And prefrontal cortex is actually connected with all other cortical region, and that way it might change the processing to the whole brain. So this selective alteration of these neurons, which are during development, so meaning their development, it's present during age 2 to 5, and all other neurons are already at the fully mature stage regarding dendritic development and their size. These neurons show development, show further dendritic growth, as you can see here. This is one of our major findings, which we of course connect. So you can see here that most of other neurons have reached by age of 1.5 and 2 the dendritic complexity, but these neurons will continue to grow, which we connect to the development of intracortical connection. Their synaptic development also show that they have, especially the parts which are distal oblique dendrites, which are involved in cortical processing, that these parts has the most protracted and the highest synaptic overproduction, which extends up to third decade of the life, and which shows the huge plasticity of these neural circuits up to the third decades. So when we were looking, because this research were also including some subjects which are corresponding to the old age. So when we are looking to alterations in dendritic structures, we couldn't see that these neurons show really some changes. But when we are looking to spine, we see that some of them preserve their synaptic density, but we can see still this decrease. This in part of the subject decrease in the synaptic or spine, meaning synaptic density. So when we compare the adult with aging on different parts, we see, especially on some parts, clear tendency. It was not statistically significant, but it was around the level of the significance that there is a decrease in the synaptic density. As we mentioned, when we connect development and aging, we have to keep in mind that everything what happens during the fetal life, during the postnatal life, and these neurons show high plasticity for a very long period, might be reflected on the aging, even on the aging process, not only on the final neurocognitive outcome, then also on the aging process. And finally, at the end, I would like to thank my collaborator, which are co-authoring this, also this study. Many of them during past years reached to become assistant professor. Some of them are on the way to achieve this level very soon. And these are our PhD students, which are the best students of the generations. And we are very proud that we can have it on Department of Anatomy and working in this research. Thank you very much.